Peroxisomes are single membrane bound sacs filled with special oxidative enzymes and a crystalline core suspended in the fluid. Peroxisomes convert toxins and poisons in the cell into hydrogen peroxide. They also aid in the digestion of long-chain fatty acids. Hydrogen peroxide itself is a harmful substance and must be disarmed immediately, which is also done by the peroxisome, specifically the enzyme catalase, which is contained in high concentration inside the peroxisome. The hydrogen peroxide is then turned into water by catalase, and oxygen is also a byproduct of this reaction, effectively ridding the body of toxins. These organelles are found in abundance in liver cells of mammals because it has to process alcohol and other toxins. Hi, I'm Greg Smith, and I am in the first period AP Biology class at Creighton Prep with Mr. Fife. Today I will be demonstrating peroxisomes' interaction with hydrogen peroxide, which they create in their membranous enzymatic fluid. So the way I'll be showing you this is through hydrogen peroxide and beef liver, which I have here on a plate. Liver cells are particularly high in hydrogen peroxide and peroxisomes because they deal with toxins and getting rid of alcohols and that sort of thing. So liver has lots of peroxisome in it, and peroxisomes create hydrogen peroxide to destroy toxins that would be harmful to your body, and then enzymes inside the peroxisomes, catalase specifically, gets rid of the hydrogen peroxide because it is also poisonous and toxic. So then they take the hydrogen peroxide, which is broken down by catalase, and turn it into water and oxygen. So we will see that reaction today. Alright, let's get started. First thing you're going to want to do, take your hydrogen peroxide and pour it into your glass beaker, whatever you're using. Throw that in there. Looks good. It actually looks like water. Don't drink it though. That would be bad. Next, you're going to want to add your beef liver. This isn't a really complicated process. Pretty easy, actually. Now, let's add your beef liver. Make sure to cut off a piece that's not too large or too small. Here we are. Beef liver to hydrogen peroxide in 3, 2, 1. As you can see, the reaction happens almost immediately on contact. Right now, the peroxisomes are using their enzymes to break down the hydrogen peroxide, which is toxic in humans, to water and oxygen, which is the gas you can see it's releasing. It's a relatively rapid reaction. There are hundreds of thousands of enzymes in this beef liver tissue, and it will completely react with the hydrogen peroxide until nothing is left but water and oxygen. And, well, there will be beef liver in there too, but, not the point. So that's how that looks. It's really quite fast. The reaction is exothermic. If you feel a glass, you'll notice it's a little bit warmer. Not scalding hot, but it's definitely warmer. Peroxisomes are budded off of the ER and are located in the cytosol near the ribosomes due to the lack of peroxisomal proteins. Peroxisomes need to have their proteins imported, and being near the ribosomes allow that to happen. They're also located near the mitochondria and chloroplast because of the role they play in digestion. In chloroplast, peroxisomes aid in photorespiration, and mitochondria, they aid in the Krebs cycle. Without peroxisomes of the cell, we would not be able to rid our body of toxins, and our cells would die. One interesting fact is that peroxisomes may divide to create two new peroxisomes without DNA. Ribosomes are produced in the nucleolus, a prominently visible body within the nucleus of eukaryotic cells, which is an aggregate of ribosomal RNAs, ribosomal proteins, and other macromolecules held together and organized in a way not currently elucidated by careful scientific study. An easy way to think of this is a body comprised of ribosome parts. These pile of parts magically create ribosomes. The ribosome consists of 50-something proteins and a stack of rRNA. The overreaching structure is one of two subunits, one larger and one smaller. The smaller subunit functions to bind incoming mRNA. The larger subunit serves as a housing for three chambers through which tRNA is shuttled. It also houses the peptidyl transferase site, which allows the transfer of the amino acid from being bound to the tRNA molecule to being affixed to the elongating protein chain. Ribosomes are essential to cell life and are used to synthesize proteins. This process is known as translation. They are translated from mRNA and are mediated through the tRNA. This is called translation because RNA translates from genetic codons in the mRNA to the protein monomers. Amino acids are the subunits of proteins and polypeptides. Ribosomes snap these subunits together in an appropriate order, forming peptide chains which fold either on their own or with the aid of chaperones into the appropriate secondary and tertiary structures. These then will proceed to create secondary, tertiary, and finally quaternary structure. The ordering of these monomeric subunits is the primary structure. Ribosomes are located throughout the cell and aid in many cellular functions. They interact with the endoplasmic reticulum, the rough ER, and are produced by the nucleolus. Even though this is true, they aid almost every single membrane-bound organelle by creating proteins that help in their function. The importance: without these ribosomes, there would be no life. Life would be impossible. Protein synthesis is what makes us able to live.
What do we know about partying or anything?